Okay, Assalamualaikum. We are now in the part 2 of Neoplasia 1 lecture. Okay, we have done already these three in part 1. So now we are going into the part 2 and part 3, which tells the story of the hallmark of cancer. Okay, hallmark of cancer by definition is just the fundamental changes and in the cell physiology of a cell, okay, from a normal cell into a malignant cell. Okay, so basically there's actually if you look into your textbook, you'll find there's 10 of it. Okay, 10 of features, but which consists of number 1, sustaining proliferative signal, number 2, evading growth suppressors, number 3, deregulating cellular energetics, number 4, resisting cell death, number 5, enabling replicative immortality, number 6, inducing angiogenesis, number 7, activating invasion and metastasis, and number 8, avoiding immune destruction. However, there are two other features, A and B. Okay, they put it together in the hallmark, but these are actually enabling characteristic. What does enabling characteristic means? Is actually features that promote cellular transformation and subsequent tumor progression. So basically, all other characteristic is actually the main characteristic, and these two are the one that helps the characteristic of the hallmark of cancer. Okay, the hallmark of cancer is divided into two parts. The first part is the one that I'm doing now, which consists of self-sufficiency of growth signals, insensitivity to growth inhibitory signals, limitless replicative potential or immortality, as well as evasion or apoptosis. Mind you, I'm not going to give you a very detailed explanation on each particular genes that is involved. I'm going to use them as an example only. So this is just an overview of all this hallmark for you to remember. The one that of all these four in part A, this is the most important one. Okay, in part B we'll go into the rest later on. Okay, now first of all, in self-sufficiency of growth signals, where does this originate from? It is basically from gain of point mutation. What happened is actually in a cell there's actually a nucleus, and in the nucleus there is actually DNA. Okay, so what happened is there's actually some mutation that occurs in the DNA that convert proto-oncogenes to oncogenes. Okay, what are proto-oncogenes? This is normal occurring genes that in the normal function of the cell. And what is oncogenes? Oncogenes is abnormal genes that encodes proteins called oncoproteins. So what does this means? This means that these proteins then produced by oncogenes will promote cell growth even in the absence of normal growing promoting signals, meaning that for cell to grow it needs some stimuli, but when there's actually oncoproteins, the cell just grow without any stimuli. There's no need of it. Okay, now in a normal growth of cells, growth factor is very important. Okay, so basically the process involve five processes involved. Number 1 is binding. Okay, the growth factor binds to the receptor. Number 2 is activation, the activation of the growth factor receptor. Number 3 is the signal transduction. Okay, from the growth factor before it goes into the nucleus, there's actually transmission that may occur, and when it's inside the nucleus, there is transcription. Okay, there is transcription of the signal to another DNA to produce something else for growth, and mainly is for the cell to enter the cell cycle. So this is cell cycle. Okay, Now, in terms of if we look closely, you can actually see this is the binding part, this is the activation part, this is the transmission part, and this is the transcription part, and that's the entry part. Now, in the binding part, the problem is the growth factor availability. Okay, so what happened with the growth factor? What's abnormal in cancer cells growth factor? The cancer cells can secrete their own growth factor, number one, or they can induce growth factor production by the stroma. Okay, this is a stroma. Okay, let me show you. All right, remember this. Okay, so what happened now? The tumor cells. This is tumor cells. What they do is they actually produce a growth factor inside the cytoplasm, and this growth factor will go outside. Okay, out of the cell, and then attached to the receptor. This is the receptor. Okay. Second thing that they can do is actually the tumor cells can actually produce cytokines. Okay, the cytokines goes into the outside of the cells. Okay, and so-called stroma, and then this stroma produce a growth factor, and again this growth factor will attach to the receptor. So this is how abnormal tumor cells or malignant or cancer cells do to make sure that they can actually live without a growth factor that is actually produced by something else. They themselves produce a growth factor. Okay. Second part is activation. Okay. In growth factor receptors activation, the problem is either mutated gene that causing increased expression of the growth factor. Okay, the gene in the cell, there's actually nucleus, there's chromosomes. The gene is mutated, so what they do is they produce a lot of receptors. Okay, and mutated growth factor that causing automatic tyrosine kinase. Okay, this problem with the slide, kinase activation. What does tyrosine kinase activation means? Tyrosine kinase activation always means there is actually proliferation. Okay, so that's important, right? So example of increased expression, the normal breast cells, epithelial cells, there is this number of receptors. Okay, 25,000 to 50,000. But when this part of the DNA is mutated, what they do, they express a lot more. You see, there's only probably in one surface 1, 2, 3, 4 receptors. But here, the receptors is much more, and it's said to be 10 to 100 times more. Okay, so when it is 10 and 10 times 100 times more, there will be excessive cellular division, because there is no limitation of how many growth factor that can attach to it, and the cell will grow and grow and grow and grow without any stop. Okay, next is the tyrosine kinase activity. Example in EGFR. A normal EGFR, this is the receptor of EGFR. A normal EGFR needs to be combined into 1 and 2, and then only the growth factor will attach to the to the area of the protein and cause the production of signals, and this will stimulate the tyrosine kinase activity. Okay, so they stimulate tyrosine kinase activity into proliferation. But in malignant or cancer cells, the DNA there's mutation in DNA of in the chromosome of EGFR that produce this type of EGFR. So what does this EGFR do? This EGFR are mutated protein, okay, protein as a growth factor at the membrane of cytoplasm, and what does it do is automatically produce signal to stimulate tyrosine kinase. Okay, so therefore all of this will cause proliferation, okay, or tumor, right? Next is another example that they use in the book is BCR-ABL protein. This is not in the receptor, okay. This is basically a protein there is no abnormal BCR-ABL protein. The normal one is only BCR protein. Okay, this is basically a protein that used for proliferation as well, normal proliferation. However, this protein is produced. This protein is produced by chromosome 22. However, there is some problem in some patient that has cancer. There is translocation of chromosome 9 to 22. They produce a new chromosome, and this new chromosome will produce this protein. Okay, with this protein combined to what they will produce is actually more production of this proliferation. Okay, there will be more proliferation, right? So how cause more proliferation? This protein can actually go into the cell membrane, and it can actually become a receptor to produce tyrosine kinase activity to be increased. Okay, right. Next is transmission. Okay, we're done about the growth factor. We go to the transmission part. Now, in the transmission, if you remember, after the growth factor attached to the receptor, and then it starts to have some transmission process before it goes into the nucleus to become to go into transcription. This whole process. There's two main protein that they use in the textbook. Number one is RAS and BCR-ABL. Again, the same thing that you have seen before, but this one is in terms of downstream protein. Now, what is the problem with RAS? Number one, there's mutation of the gene producing abnormal protein that causes abnormal GTP hydrolysis. Okay, I'll show you later on, and also the mutation of enzyme that hydrolyze GTP. Okay, in BCR-ABL, basically the fusion of gene just now. What does it do? That particular protein, not only in the receptor, it causes problem in the cytoplasm itself. It can cause more the transmission to be more than usual. Okay, more transmission, so therefore it will produce more cells. Okay, produce more proliferation signals. Now, what happens in mutation of gene producing abnormal protein? Okay, in normal tyrosine kinase activation, okay, there is an inactivated RAS become activated RAS, and then this will then produce proliferation. Okay, it will proliferate. Alright, but this is actually short-lived. Okay, because there is actually enzyme, hydrolysis enzyme, that actually cause the activated RAS to become inactivated, so it will stop the production. But there is a problem with this pathway. So there is no inactivation of RAS. So what happens is, when there's actually mutation, this particular process does not occur, and the RAS keep going on and on and on, and produce more proliferation, and proliferation. Okay. Another thing is, before RAS inactive become active, it needed from active to inactive, it needed an enzyme, GAP enzyme. Okay, these GAP enzymes can be mutated also. Right, it become mutated, therefore there's no GAP enzyme. So what happens is only activation, and again it causes more proliferation and proliferation. Okay, right. So that's the mutation, right, causing that, and again mutation. Okay, in terms of transcription, okay, remember just now the receptor, right, the transmission, okay, and then now it's in the nucleus, it goes into transcription. Okay, right, transcription. So in transcription, the example that they use in the textbook is MYC. Okay, what are the problems with the MYC gene? Is there is an abnormal expression of growth promoting genes, okay, as well as that will cause the promotion of progression into cell cycle. Okay, and then another one that can occur is the dysregulation of MYC gene will cause enhanced alteration of metabolism that support more cell growth. Okay, So what happened? Okay, this is actually the transcription that occurs in MYC genes. Okay, in any receptor activation. This is the transmission, and this is the transcription. The MYC gene is the one that transcript the signal to make sure it goes into cell cycle, it goes into apoptosis, it goes into proliferation, it goes into metabolism, and it goes into production of more RNA. However, this is controlled. A normal MYC gene means that it control the whole thing to a certain point of time. Okay, when this MYC genes become abnormal, what happen? This thing become more and more. Okay, so there will be more cell cycle, right, but not more apoptosis. It will less apoptosis, more proliferation. Okay, mainly these two things. And again, in this thing, as shown in that box, it will cause increased metabolism. Okay, so that's how MYC gene, the normal one, supposed to control this, but when it becomes mutated, all this process become enhanced. Okay. As shown is here, this is all the things that normally MYC gene will do. Okay, everything that related to increase growth. Okay, but when there's actually dysregulation, okay, as well as amplification, this thing becomes abnormal. Okay, so it will become more signal transduction, more cell cycle, more metabolism, more translation. Okay, and it will cause more growth. Okay, and the last one on the self-sufficiency of signal growth is on the entry. Okay, the problem is either there is actually gain of function mutation involving CDK4 or D-cyclines, as well as loss of function or mutation involving inhibitors. It's a bit complicated, but look at this. Okay, I think you remember that this is what we call as cell cycle. Okay, so in cell cycle, okay, there is many entry points. The main entry points for cell to go into proliferation is G1 and S. Okay, but within the cell cycle, there's a lots of checkpoints. Okay, and the one that actually makes the cell goes into the cycle is these two things. It is called as inhibitors and promoters. Okay, there's different type of inhibitors: CD4K, CD46, CDK6, CDK2, and also the different cyclins. Okay, different cyclins D, majority of cyclins D, and other cyclins as well. So what happens is when there's actually gain of point mutation, meaning that there's actually more function of the cyclins, D cyclins. Alright, okay, when more function of the cyclins, this will go even more, okay, towards the cycle. And when there's also mutation inhibitors, meaning the inhibitors is supposed to check, there will be none of them. Okay, when there's no check, the checkpoint will not work. When it does not work, it will go on and on and on and on and on, and the cell, it will proliferate and proliferate and proliferate like mad. Okay, so that's the problem with entry. So we have done with the self-sufficiency of growth signals. Okay, second is insensitivity to growth inhibitor signals. Okay, in cell growth, okay, there's always growth inhibitors as well as growth factors. Okay, please disregard this. Okay, just look into this. This whole process is a process to make sure that when the cell is growing enough, there will be transcription to make sure that the DNA, the cell stop from growing anymore. Okay, how it is done? Usually, there's actually a protein called RB protein. And this RB protein will actually become inactivated, and this inactivated RBGs are hypophosphorylated. These are the phosphate subunits. It become hypophosphorylated. Then when it becomes hypophosphorylated, it will form this particular complex, and this will cause transcription block. Okay. However, in cases of cancer, there's a mutated RB gene. What happens in mutated RB gene? All the RB gene become hyperphosphorylated. When it becomes hyperphosphorylated, it will release this type of protein, and this type of protein will cause transcriptional activation. This will mean that there will be more and more and more proliferation. Okay. Number 3, limitless replicative potential or immortality. Okay, as you have learned before in the cell senescence lecture, cell is supposed to die. Okay, it's supposed to become old, and later on it will die. Okay, so what controls this? It's because of the telomeres. Okay, so in a normal cells, the telomeres is inactivated and later on become shortened. At time it starts to becomes old, and later on it becomes old. Okay, all right. So the problem with cancer cells is there is actually some genes that actually blocks the shortening of the telomeres. Okay, the telomeres is not shortened; the telomeres become prolonged and prolonged. Right, and then this is the prolonged telomeres, then that will cause the cancer to be not dead. Okay, so the cells rather than normal cells, it will always be replicating, but there is to make sure that there is enough space. There will be some cells that supposed to be old and dead, and it will be replaced by the new cells new cells later on. But if this cell is not dead, it become immortal, then that's what we call that a tumor. Okay, it becomes non-replicative. The old cells stay, the new cells still grow, and this is what the whole thing is called a tumor. Okay, and the last one for this part is evasion and apoptosis. I hope you remember already what are the process involved in apoptosis. Okay, that makes a cell to produce apoptotic bodies. Okay, not completely dead, but in this process, okay, the normal process that's supposed to control this are actually these two genes, BCL2, BCL-XL, and MCL1, as well as the p53. So when there's DNA damage, okay, cell is when there's actually damage to the DNA, cells are supposed to die. Okay, but this is actually helped by the p53 and the BCL2. However, in this case, apoptosis does not occur because these genes are mutated. Okay, when these two genes are mutated, there's none of these genes. This it's not none. It's basically non-functioning or abnormal functioning of those genes. Then apoptosis does not occur. Okay, so it will go into again same thing or similar to the immortality. The cells will grow and grow and grow and grow, form a tumor. Okay, I think that's all. We'll continue later with the part 3, the hallmark of cancer, part B. Thank you.